The topic of my presentation is the effects of zinc, manganese, and taurine supplementation on eggshell quality in after peak production high-laying hens. I'd like to start with a brief introduction. As you know, eggshell consists of 5 distinct layers: shell external, shell internal membrane, external shell membrane, placid layer, crystal layer, and cuticle. The quality of eggshell is related and associated with the quality and integrity of these distinct layers. From economical and public health point of view, eggshell quality is important, and as you know, approximately 10% of laid eggs has been broken during gathering from farms, transferring to the market, and therefore approximately 10% of eggs annually has been lost. From public health point of view, when eggshell is interpreted or eggshell has broken, some microbial infection can occur, and for example, salmonella infection of eggs is happen. But take a closer look on factors that can affect eggshell quality. An important factor that determine the eggshell quality is mineralization and the height of the placid layer of eggshell. Placid layer is the highest layer of eggshell. Some factors that can affect the placid layer height are microminerals and vitamin D6. Microminerals such as calcium and phosphorus, which are supplied by diet, can affect the mineralization process of eggshell. Another important factor that can affect eggshell quality is eggshell matrix proteins. Matrix proteins consist of 2 classes: calcium binding proteins and proteoglycans. These proteins have an important role in the integrity of eggshell and can determine the eggshell strength and mechanical properties of eggshell. As you see in this picture, there are many proteins that are directly involvement in eggshell mineralization. These proteins can arrange calcium columns and can form the calcite columns in the C-axis. Therefore, they arrange the formation of calcite axis. For example, in this picture, you can see that one of the eggshell proteins can arrange mineralization sites or mammillary knobs on eggshell membrane, and therefore they can manage the structure of eggshell. The third important factor is eggshell membrane and mammillary knobs. As you know, mammillary knobs and eggshell membranes are consist of disulfide-rich proteins fibers, which approximately contains 10% cysteine, collagen X, lysyl oxide, and lysozyme. Totally, mammillary knobs provide a temple for mineralization, and they provide the sites for calcium carbonate crystals, which you can see mammillary knobs on the eggshell membrane in this picture. There are many research that focus on the role of microminerals on eggshell quality in layer hen, especially in after peak laying hen. For example, manganese and zinc have been researched by some studies, and it has been shown that they have determinant effects on eggshell quality. For example, manganese has participate in protoglycans and glycosamine glycans synthesis. It can create a rigid frame of minerals that providing more resistant and strength for eggshell. At the other hand, zinc is a component of carbonic anhydrase enzyme, which supply carbonate ions for eggshell formation. Also, it can increase mammillary corn width and the expression of mRNA of proteins which are involved in eggshell formation. But another important micronutrient which have received more attention is taurine. Taurine is an non-proteinous amino acid which has not participate in protein formation. There are some studies which evaluate the effects of taurine on laying hen performance. For example, it has been shown that taurine can maintain the eggshell gland integrity and oviduct from injuries and inflammation in laying hens. Also, it can regulate the calcium hemostasis. For example, it can control the calcium bioavailability. When the calcium concentration is low, it can increase the calcium concentration. And when calcium concentration is high in uterine fluid, it can reduce the bioavailability of calcium, and therefore it regulates the calcium hemostasis. Also, it can inhibit the expression of collagen X in eggshell membrane. But what is collagen X? Collagen X is a hypertrophic agent for chondrocytes and provide a boundary for biomineralization in eggshell membrane. Therefore, the formation and the structure of mammillary knobs in eggshell membrane is under the control of expression of collagen X. At the other hand, it has been shown that supplementation of taurine in laying hens and quails diet can increase the density of calcium calcite columns. With this introduction, we conducted a study to examine the effects of these two micromineral, zinc and manganese, and taurine on the eggshell quality of laying hens. We adopted 70-20 laying hens at 70 weeks old. 18 treatments had adopted in a completely randomized design and a factorial design. A corn soybean meal basal diet were used with no supplemental zinc and manganese. The duration of experiment was 14 weeks, 8 weeks for adaptation and 6 weeks for experiment. In this table, you can see the supplemental levels of zinc, manganese, and taurine in the on basal diet. Zinc provided from zinc oxide, manganese provided from manganese oxide. The final concentration of zinc and manganese and taurine in diets was according to this table. At the end of experimental period, we select 6 eggs from each replicate, and the eggs weight were recorded. Eggshell thickness was determined by a micrometer, and eggshell mechanical resistance and eggshell static strength were examined by these procedures. The results of experiment has shown that most of eggshell quality criteria at week 76 has affected by had affected by the interaction of manganese and taurine. For example, shell percentage, eggshell thickness, and eggshell strength, and also eggshell mechanical and static strength were affected by dietary treatments. However, the main effects of zinc and main effects of manganese and taurine on some traits were not significant, but the interaction of these micronutrients could significantly increase the eggshell quality criteria. For example, the interaction of manganese and taurine has improved eggshell static and mechanical strength. However, the eggshell thickness was not affected by dietary treatments. The interaction of zinc, manganese, and taurine could only affected eggshell mechanical strength. Totally, the results of the current study was in accordance with some other researchers who have shown that eggshell broken strength affected by zinc and manganese, while eggshell thickness was not affected by dietary treatments. In these pictures, you can see the effects of different dietary treatments on the microstructure of eggshell via scanning, microscopic scanning, electron microscopic. As you can see in dietary treatments which have received taurine and manganese in comparison with control treatment which had no supplemental taurine, manganese, and zinc, there is a difference in terms of the structure of eggshell. For example, the density of mammillary knobs and calcite columns is more and higher in treatments which with supplemental taurine and manganese compared with those which with no supplemental taurine in these pictures. At the other hand, the integrity of pellucid layer in the treatments with supplemental taurine is more evident and clear than those with no supplemental taurine and manganese. It can be concluded that the effects of taurine and manganese on eggshell quality and eggshell static and mechanical strength is apart from eggshell thickness, because however the eggshell thickness was not affected by dietary treatment, but the integrity of eggshell structure and the mechanical and static strength was affected by dietary taurine and manganese and improved. We can relate this observation to the role of taurine and manganese and especially taurine on eggshell matrix protein and protoglycans. As you know, matrix proteins can regulate the formation of calcium columns, and therefore they can affect properties mechanical of eggshell. In this slide, you can see the effect of taurine on the structure, microstructure of eggshell. For example, the height of pellucid layer in taurine supplemented treatment was higher than treatment with no supplemental taurine. At the other hand, the height of mammillary knobs and mammillary layer is lower than the treatment with no supplemental taurine. It can be concluded that taurine by affecting the density of mammillary knobs and by affecting the number of mammillary knobs and affecting the density of calcite columns can provide a more integrated pellucid layer, which directly can affect the mechanical and static strength of eggshell. Totally, the results of current experiment have shown that taurine supplementation in combination with manganese can improve eggshell strength, and it can adopt it as a strategy to improve eggshell quality. At the other hand, the effects of taurine, manganese, and zinc on eggshell quality is dependent of eggshell thickness and might be related to the eggshell matrix structure. Thank you for attention. If there is any question.